Good afternoon, everyone. I hope I'm audible. Can someone confirm once, please?
Yes,
Yes,
sir.
I hope you have learned this one particular poem, or you heard about this in sometime in school by William Blake, Tiger Burning Bright. I just picked it up to mention. In the last class, we were discussing about the story of lions and how nature works with the twisted story, and that there are many things about it, that infanticide, killing, and mating, and many other things. This is just one about one species. And if you take about predator, prey, and so many other things, it is a whole new level of drama that happens in this on this episode. I like this poem that from William Blake. You are encouraged to read that. But uh, I want to go to here. I mean, if you see, what shoulder, what art could twist the sinews of thy heart, and why that heart began, did the hand, what dread feet did they feel afraid? The most striking of it is, in a philosophical sense, and the beauty of the words and the poetry that they put in. Did he who made the lamp make thee? Right? I'm trying to again just use it to say how much drama is there on the greatest show of on earth, is the fight between different organisms, right? You may want to also have a look at it sometimes. Okay, I hope in the last class, the essence of the last class is to say evolution and nature. Nature is the engineer that will allow the strongest to survive, and the others will perish. When it comes to humans, that cannot be said the same. We are intellectually superior, and we are a huge gathering. That means we are masses of multiple brains, and that means we can do many things much more better. We can override the selection pressures that the nature put on us. If it is cold, we will light a stake and we will get the heat from there. If it is too hot, we will construct a house and be in there. If there is no water, we'll drill a bore and we'll get water out. We have solutions for all the kinds of stringencies that the nature may have for us. So we are no longer under natural selection, not as much at least. And then that will make the problem of the infiltration or penetrance of bad genes into the population. Whichever is coming new with the mutation will accumulate those mutations in the next generations, because we have ways of circumventing the diseases itself, right? So what that is we discussed all these things to build, and as to build, why should we do anything at all? Why not leave everything, all of it, and live as we exist already without any improvement? What will happen? Our human species might have serious trouble in that case. So, what we are going to do is today we are going to learn a little bit about cutting and joining the DNA. Before that, we will have an overview of GenTech engineering, and then we will go and discuss a few more things. In the last class or so, I described about genetic engineering in the nature. Nature does it to make useful traits that will allow. I mean, I'm using anthropomorphism, but otherwise, those that are fit will survive. The rest will die. So best engineered ones will survive. The poor ones will die. And here we are doing it artificially. We are engineering genetic circuits or gene circuits such a way that they will give products or consequences of our interest, and that is the essence of genetic engineering. In all of genetic engineering, we have a purpose, something like in majority, if not all, we have, say for example, we have a DNA molecule, something like this, and we have another DNA molecule, something like this. And then we are supposed to combine these two and make one DNA molecule. It can be many other possibilities are also there. We'll just take the simplest joining of these two. And now we made a recombinant DNA. Right? This course is rDNA, recombinant DNA technology in medicine. So our applications will be mostly of medicine, but otherwise, essentially, we are reading genetic engineering. So the overview is to engineer such things, we should have a ways of cutting the genes. First thing, of course, is to identify the genes of interest that will give the product of our interest, or to make the gene of desired with desired genetic information. So we should do that, and then we should put it into a vectors. Those are the other DNA or nucleic acid based molecules that will carry the gene of interest or harbor the gene of interest. And then we have to mix them up, and then we come with to recombinant DNA molecule. That recombinant DNA molecule could be put into a cell, and that is only when the replication can happen, and also the expression. Through replication, you will make multiple copies, that is cloning, and through expressing the genetic information on the vector or recombinant DNA, we will have products of our interest or phenotypes, right? So that will be the essential part of this course. And but each of these steps will have multiple techniques or methods. Say isolation of gene can be done by PCR, constructing genomic libraries or cDNA libraries. Vectors will have a whole huge 3 or 4 classes just on vectors. And we will learn about the intricate details of them, plasmids, cosmids, phagemids, and so on. And the recombinant DNA, whether we are making it chromosomal or extra chromosomal DNA. We will know about these two, and we will learn a little bit about the transformation, transfection, biolistics, and eventually we will also learn about sequencing, protein engineering, etc. A little bit of all. And then we will see some applications that are done in the field of medicine, which could include diagnosis, vaccines, or treating, reprogramming, cell reprogramming, and so on. The first thing, however, is we need to make a recombinant DNA. If we want to make a recombinant DNA, we should have cut the DNA, pieces of DNA, and then join them. And today's class is about how to cut the DNA, right? And that is using restriction enzymes or endonucleases. First, we will learn about the discovery of restriction enzymes. It will get a little bit complicated. If you don't understand it today, it is all right. You can watch it multiple times, or you can come up with specific questions, and then we will discuss about it. Why I want to have the discovery of restriction enzymes? It's over, it's kind of history. Why do we need to know it? I'm not bothered about the point of restriction enzymes. I'm bothered about how people found it out. Why did people find it, and from what indications, and how they have gone through it. Mostly in science, we learn the science part of it. We forget to read the history of how those things have happened. Because why that is important is we will come across many things that will go about like the penicillin, for example. The discovery of penicillin. The bacterial culture was there, and in one place there is fungi, and around that there is no growth, but everywhere else there is growth. If we find any plate like this in our lab right now, we just discard and throw it away. But that Alexander Fleming, he didn't do that. He was wondering why did something like this happen? Why didn't they grow around it? And then pursued, and now we have the whole antibiotics. So I would want you to also, for that reason, I would also want you to learn about a little bit of history and try to know how they have progressed through and found out. And I hope you know that there was Nobel Prize given for discovery of restriction enzymes. You all must have studied enough, at least the biotech students must have studied about bacteriophages. And even bioengineering in molecular genetics or molecular biology, you have read about lambda phage, right? Lambda phage infects bacteria, and here we'll just talk about general lytic cycle. They infect bacteriophages infect bacteria, and they deposit their genome, or they inject the genome into the bacterial cell, and that bacterial genome will produce more copies of itself and the capsid proteins. Capsid proteins are the surrounding cells, surrounding proteinaceous coat around the genome. The purple one is the genome here. So it will produce more of those, and then the new phages are assembled, and then from inside they would lyse the cell, and new phages are released out. That is a reproduction cycle of phages. It's lytic phage because at the end they lyse the bacterial cell and then released out. The opposite in a way is lysogenic. I'm not going to discuss this at this point. Lysogenic life cycle, right? This is what is happening. If you see it on plates, you can have a lawn of bacterial cells. So you take, I'll just describe briefly. So you take a petri dish, you put a layer of a layer of nutrient agar, and then you pour a little bit of soft agar, which is little less dense or mixed with E. coli or some bacteria, say E. coli. So because of which I'm drawing the cross section of the plate, so the bacterial culture will grow all over it. But if you have mixed a little bit, a few numbers of phages, okay, phages, what would have happened is that in that location, say, let me try if it works. I will just draw the whole plate like this, okay? So if you had phages here, that phage would have infected one bacterium, and upon lytic cycle would release more phages, and they would infect the other bacteria, and they would kill off all the bacterium that is around here. Okay, I have difficulty drawing it as a lawn. So this assume that this is a lawn of bacterial cells. In that place, you have this part where all the bacteria at that place are lysed by the phage. And each of those, as you're seeing here, is referred to as a plaque. It is kind of opposite to a bacterial colony, right? If you have nutrient agar and you put one bacterium, after a few hours, say after a day, you'll find a colony of cells. This is a colony of bacterial cells. Here you have the whole bacterial lawn is there. In that you have a clear zone that is shown something like this, and that is a plaque. I hope that is clear and simple enough. Now I want to describe what is efficiency of plating. Say for example, I took 4 phages, bacteriophages, and I have put it on a plate along with bacterial culture. 1 second. So what will I get in that case? So I have put 4 phages and everything done as shown here. Nutrient agar is there, and bacterial host bacterium is also there. How many plaques should it form? If you have 4 phages, each of them assume has infected one bacterial cell, and they would form a plaque. So that is the efficiency of plating is, you can think of it as number of plaques formed by number of phages added. Okay, we'll talk in, if you're saying in percentage, then you should just do that. So now I added 4 phages, I found 4 plaques, so it is about 100% efficiency of plating. Say for example, I have put 10,000 phages and I got about 20 or say 100 plaques, then the efficiency would have been 100 by, that is 10,000, something like that. It's about 1% efficiency. I hope you understood that, right? Okay. If you didn't understand, please, you are free to ask a question anytime. Just not raising hand. You can switch on your mic because I cannot view the screen. You can switch on the mic and you can ask a question. I hope this is simple enough, right? I will just to complicate it maybe. Let's do it this way. I have taken 100 phages and I have added it to a bacterial culture, which I will call it as E. coli K strain. And then it formed about 100 plaques. What is the efficiency of plating? That is 100%. Now I'm doing the same thing, but I'm going to change something a little bit. I have taken 100 phages, but I added it into a different strain, E. coli, say R strain. And I got 1 plaque. What does that tell you? Just by, say, that is 1%, of course, that's a different issue. What does this alone tell you? First thing is, if you have done the experiments, assume that these experiments are done at the equal time, at the same time, and all the materials are used or same, except there is a difference in K strain and R strain. First thing it means is the phage is able to effectively infect K strain well. That's why you got 100%. Somehow the same phages are not able to infect the R strain for some reason, and that is, why does that happen? So those at the very long time ago, when people were working on these phages and plaques and these things, they stumbled upon something like this. And there is a longer story to go, and this is just the introduction about it. And they were puzzled about why is that one phage is able to infect one strain of E. coli very well and failing to infect another one as effectively. Okay, so this is the actual experiment that they have done. I can't remember the names of the scientists, I apologize. The experiment is something like this. We will start somewhere here. Okay, so they have taken E. coli strain. I will put it as something like equations. They have taken E. coli K and they have infected lambda phage to this, and efficiency of plating. That means if you add 10 phage particles, then you got 10 plaques. Okay, and you would get the phages that have come about are lambda K. Okay, you call them, that means this is a lambda phage grown on E. coli K strain. That is the implied meaning there. So every time you have this lambda K and you infect on with E. coli, as is shown like this here, and you get an efficiency of plating of 1. And what they have also done is they have taken this lambda K and they infected E. coli C strain. The problem is the efficiency of, there is no problem here or two. The efficiency of plating is EOP is 1. That is 100% infectivity. And if you take, these are the phages that are formed now will be called as lambda C. Why is that? It is a lambda phage, and the C is because they were grown on host cell E. coli C strain. And if you keep infecting this lambda C onto E. coli C, you are getting EOP of 100% infectivity. The problem is if you take lambda C and infect E. coli K strain, okay, then EOP is very less. EOP is 10 to the power minus 4. That means if you add 10,000 phages, you would have gotten 1 plaque. That's something is wrong about it, isn't it? You take it again like this simple. As we saw in the previous slide, lambda C infecting E. coli C, you're getting EOP of 1. Lambda C infecting E. coli K, the EOP is very less. So people were puzzled. Say, for example, if they have added some 10,000 and they would get 1 plaque, right? Now that plaque will be lambda K. And that lambda K will show the behavior of EOP1 in all these strains. So the puzzle is this. Why is this discrepancy? If you are infecting lambda C with E. coli C strain, EOP is good. But when you're infecting with E. coli K, the EOP is less. And that was one of the first observations, and that led to, or people pulled in more people to discover what is happening and why. And what we know now is that, one second. What we know from these is that we know that some factor is actually preventing the infection of lambda C within E. coli K strain, right? If you're here, there is a problem, that is lambda K has some protection from lambda C. Why is that? How? That is one of the point. Second thing is, once this lambda C, even when it is very low efficient, somehow if one of these has formed and it is becoming, it now became lambda C, and some factor is restoring phage infectivity. Lambda K is able to infect E. coli K strain as well as E. coli C strain with same ability or infectivity. So just by growing the phage in E. coli K12, you are able to restore the lost activity. So these are the understanding or observations that led to the discovery of restriction enzyme. With using radioactivity and many other things, people have figured out a lot of things. But what we know now is that as soon as, say, assume that this is lambda C and this is E. coli K, as soon as the lambda phage DNA is inserted into the cytoplasm, there are enzymes called as restriction endonucleases that are breaking this DNA, and thereby preventing it from formation of any plaques, or they're reducing the infectivity. Okay, so if these are restriction endonucleases, which means they are endonuclease means cutting in between. exo would have been chopping off from the edges. Endonuclease, and here the nucleus is about cleaving DNA. And if this restriction endonuclease is able to cleave phage DNA, why isn't it cleaving the genomic DNA? Genomic DNA is also DNA. Both are DNA chemically. But how come the genomic DNA is safe from activity of restriction endonucleases? And that is when they figured out that there is methylation of certain sites. And they also studied that degradation is not random. The endonuclease activity, we said, it's not random. It is rather site specific. That means this endonuclease cleaves only certain sequences of DNA. Okay. And yeah, the same thing is also with methylation. And they found out that genes endonuclease, or you call it as restriction endonuclease, and methylase, they come as two gene pairs. They're usually two genes like this. And those systems are referred to as R&M systems or RM systems. This is for restriction endonuclease, which is a site-specific endonuclease, and methylase here, M for methylase, R for restriction endonuclease. M is methylase, and it is also site-specific methylase. It adds a group, methyl group, to this specific site, specific DNA sequence. So these are R&M systems. We'll have a little bit of discussion here. Sorry. If there is an E. coli strain and it has R and M, it can survive. Say, for example, I want to delete this. Okay, so that will be E. coli R minus. That means our restriction endonuclease has been deleted or made it non-functional. But methylase is functional. Can E. coli survive? Yes, no problems. If you had just E. coli and R, and if you had both restriction endonuclease and methylase, is the genome safe? Yes, it is safe.
Yes, sir.
Yes, because the genome, the sites are methylated. I'll represent like this, because of which the restriction endonucleases will not be able to cleave these sites. So it will survive. If I had E. coli, R minus, M minus, there is no danger at all, right? The danger is this one. You can assume or try to think of it in analogy like it's a sword. Okay, think of it as a sword. And you can also think of the methylase as the sheath cover of the knife. I say it's bad drawing, but I hope you can adjust with that. It's more like ice cream. Anyway, in nature, if you see, majority of the systems that come about, they always come with a protection. If something is bad, it comes with a protective mechanism or a protective protein. I will just finish this also. If there is an E. coli in which you have R plus, and R plus means present, and M is minus, then can that E. coli survive? It cannot survive, because the genome has these sites sequences which are not methylated, and the restriction endonuclease would cleave it. And once it is cleaved, the E. coli is likely to die. So,
Sir,
yes, please. Yeah, ask.
sir, sir, can you hear me, sir?
Yes, I can hear you. Ask me the question.
Sir, I'm not able to understand that. Like that, R minus and M minus could survive, but R plus and M minus is not. possible, but that those two I have doubts, sir. Like I understood for the R minus and M and R and M, like if
Fine.
both R and M are
It's
present, and
fine.
if R is alone absent and M is present, that I'm able to understand that, sir. But the last two I'm not able to get it clearly.
Okay. R minus and M minus. It's as good as it's saying that E. coli without any of these genes. There is no impeding danger to the genome. There is nothing to cleave the DNA like this because R is not there, right? If R is not there and M is not there, there is no problem at all. But if you have a situation like in the fourth situation here, where there is restriction endonuclease, but the protection is not there. If the protection is not there, then the genome is likely to be cleaved by the restriction enzyme.
Okay, sir, I understood that,
Okay.
sir. Like since like restriction endonuclease has its the property of being very site specific in terms of its cleaving. So there is also a chance that it could cleave its own DNA itself in the organism where it's isolated
That's
from.
right.
So
Yes,
that's
that's
what if there's no modification, then it will easily get cleaved, and that's what. Okay, sir. Now I got it,
that's
sir.
right.
Yes,
Yes,
yes, sir.
yes. Good. So I hope you all understood, and if you have any questions, please ask. I would be more than happy to answer. It will add a lot of value to many others. Okay, good. So I hope you understood these things. And basically, why should these systems be present even? Right? It seems like a danger, but they are present in some of the bacteria. And one of the points is that people think they pro, they confer protection from phages. What are phages? Phages are like the, say, bacteria is here, assume that, and phages are the ghosts or something that will come and keep on preying on this bacteria, right? So the bacteria needs bacteria. What is bacteria? And the essence of it is the genome here. So the bacterial genome, and here you have the phage genome. You may also have others like plasmids. There is always a fight between these genetic elements, the bacterial genome, the phage genome, and the plasmids. It's all DNA, right? If you can also think of it in the earliest ways or the primordial soup when the life actually originated. There must have been a lot of wars that are happening like this. And each of these genetic propagating elements, phage is a propagating element, plasmid is also one, and genome is also one. They are coming up with arms to protect themselves. The phage cannot survive without infecting bacteria. It has to infect, and they're only in the bacterial cell, replication will happen and then propagation. But if the bacteria gives keeps on doing that, its own survival will come into jeopardy. It will jeopardize its own survival. So it's not like they have consciously come up with these genetic elements. It's rather, it could be that they have come up, and those that have evolved these kinds of genetic systems survived better. And those that did not come up with these genetic systems must have perished. Okay, that's the basic science of it. But you should always view these things as even plasmids and phages. They are constantly um, they are what do you call parasitizing on the bacteria and trying to make use of its abilities and survive. Both the existence of phage and plasmids is absolutely dependent on the bacteria, right? Yes. So R&M systems could be providing a kind of benefit to the bacterial genome by cleaving off those foreign DNA. And at one point, we will also read about CRISPR, which is also an exciting thing. And that is one of the reasons why I'm going into this depth and trying to describe instead of just going about with restriction endonucleases. In everything that you're learning in this course, I would want you to know the finest details possible. You can actually go into depth even beyond what I'm trying to describe, but we'll not have sufficient time to do all those things. So try to be a person of details, then we will be able to make this a fulfilling course. So the rest is there are several types. The classification comes where type 1, type 2, type 3, and type 4. The difference is about the organization of, there are two differences about the protein and about cleavage. Where do they cleave? So in type 1, it is one enzyme. So you can think of it this way. R and M, they go together like this. R and M are one enzyme together. One subunit is endonuclease and the other subunit is methylase or modification, some people say. And that is the protein level. The second difference is where do they cleave? They are all these types are sequence specific or site specific. That means they recognize certain nucleotide sequences. And so cleavage, say for example, this is the site the proteins recognize and they cleave here, okay? And the distance between this is about 1000 base pairs. That's what are the features of type 1. I will repeat. Type 1, R and M are in the same enzyme, but of two subunits. The recognition of the sequence and the cleavage site is about 1000 base pairs away. Okay. Compare it with the type 2. R and M are separate. And the most important one is they cleave where they recognize. This is the site of recognition. That is where they cleave as well. And type 2 are the predominantly used ones and they're the most useful ones. Type 3 is again similar to that of type 1, where you have one enzyme and the cleavage is about 26 base pairs apart from the site of recognition. And type 2S is there, which are two different enzymes, but the recognition sequence is asymmetric. We will discuss that in a while. And there is also one other problem is that they cleave 20 bases apart. Out of these, type 2 are the best, are the most useful ones. Why is that? Every experiment that we do, should have control. We should have control about what is happening and what is not happening, what should happen and what should not happen. That means we want certainty. Okay. Say for example type 1, or I will take about type 2. Okay, because type 2, yes, type 2 is the best ones I'm saying, because you know where this site is and you know that they are cleaving it, and you don't have to add the whole enzyme like R and M together and not be sure if it is going to cleave or if it is going to methylate it. Instead, if it is type 2, you know the sequence, right? I mean, which sequence it recognizes, and you know it cuts within the sequence. So you know what are the bases here and here, and you can add what you want to add because they come as two different enzymes. If you want cutting to be happened or cleavage, then you will add restriction endonuclease, so it will cut. If you didn't want that, you would have if you wanted to protect this, you would have added the methylase so that it will be methylated, right? You are in control of what you want and you are certain of what bases will be there upon cutting. And that is why type 2 are the most useful of all these. And they don't need extra, only just ATP or so they would have needed as cofactors. If you look at if you look at type 2, yes, it is saying it is site specific. It comes in two. Say, for example, I want to use restriction. I added R. This is the sequence of recognition, and cutting is made here. How do I know what sequence is here? It is there is uncertainty. Means you don't know if it is 20 base pairs or you don't know if it is 19 or 18. We will not, we will, this is adding uncertainty. And then to go forward with experiments or any preparative techniques will become very difficult. And that's why type 2 are the most useful restriction endonucleases. And further, I mean, the advantages are summarized here. They are separate enzymes, restriction and modification. They do not require any cofactors. They recognize symmetrical and sequence and cut within within the symmetrical sequence. And some of them make staggered ends. Some of them make uh, blunt ends. Staggered ends are better. We will discuss the staggered ends in the next slides. So there is a nomenclature is something, the names are like this. They are written as first alphabet as capital, then two small alphabets and one Roman number. So S, M, A, 1. S is for genus of the organism from which it is isolated. M is the species, and the Roman number 1 indicates if it is first enzyme or second enzyme, right? Serratia marcinensis, first enzyme. That is how it is, SMA1. Haemophilus aegyptus, third. Then it is HAE 3. Hind 3, Hind 2, Haemophilus influenza 2. In some cases, you will also have a strain name. Say, for example, BAM-H1, BAM-H1, Bacillus amyloliquefaciens, and strain H. We didn't have, in all these, we didn't have any alphabets in the Roman numbers one. Here you have H1, which is strain H and first enzyme. And in the olden days, this is compulsory that we have to use italics. Only in the last year, I came to know while teaching this course from some of the students that italics are no longer required. So you can also Google about it, okay? But I hope you will remember this. Nomenclature includes genus, species, strain, if it is available. Not all are, say here, for example, there is a strain D, but it's not mentioned. Some of them, they have it, so we'll just follow that. And number of the restriction RM system identified in that organism. 1, 2, 3, and so on. There is also one more. So SMA1, if I write UR meaning restriction endonuclease. If you want to make M methylase, then you have to write M SMA1. That would mean that it is a methylase. All properties similar, except that it's not going to cut the DNA, it's going to instead methylate. The top one should have been something like this, but we don't have to do it. Just any of these, all the ones that are indicated here are restriction endonucleases. Only occasionally we use methylase, and in that cases you should have mentioned as M-BAMH1, for example. M is for methylase, bacillus amyloliquefaciens, H strain, and serial number, that is 1 of the discovery of the restriction endonuclease. So there are hundreds of restriction endonucleases that are identified in various bacterial strains. If you go to NEB, New England Biolabs website, they have a whole repository of giving their properties, sequences, and many other things, many other details that could be read before you order them. So some of the examples are EcoR1 and BAMH1. I don't want you to mug up all these restriction sites except for BAMH1 and EcoR1. GAA, TTC, GGA, TCC. Why I want you to remember at least these two is these are the only two restriction endonucleases I remember. Okay, otherwise there is actually no reason. Um, and in many of the examples that we will discuss throughout the course, I would tend to use these two often. And then it becomes easier if you already know. And the difference between these two is not much. GGA, TTC, GGA, TCC, right? And restriction endonuclease can also be categorized on the number of bases they recognize, the sequence, the site, number of bases within the recognition site. Majority of them are 6 base cutters. That means they recognize 6 base pairs, bases, and then cut within the site. Some of them are 4 base cutters and some of them are 8 base cutters. There are also variations, but we don't have to worry about it. Yes, so I will go into the sequence recognition sequence, the architecture of the sequence itself. So they are a palindrome. I think these are very easy for majority of you. That is, you read from here to here, or in the reverse direction, you will read the same. A palindrome is something like this. Abel was I ere I saw Elba, supposedly said by Napoleon when he was imprisoned onto an island and he was looking at another place also called as Elba. Abel was I ere I saw Elba. It's a good palindrome. A, A, B in the spelling wise. If you see, you are seeing the L, L, E, E, W, W like that, right? If you are reading it in alphabet wise, you read from in this direction or you read in this direction, you are reading the same, right? In DNA, what is a palindrome is you're reading, always reading should happen 5 prime to 3 prime. There is no question about it. Even mRNA or DNA or RNA, every reading of nucleic acid should happen from 5 prime to 3 prime. So when you're reading a double-stranded DNA, you read GAA TTC. And when you're reading the lower strand, you read from this direction, GAA TTC. And there is an axis of symmetry, right? This half, this half is similar or identical to this half, right? That is the axis of symmetry. So basically, the restriction endonucleases in type 2, they form a dimer like this. This is cyan in one dimer, one subunit, and the other green in other subunit. They bind to the DNA that is shown like this, and one of the subunits make a cut here, and another one makes a cut there. So all the restriction endonucleases that we have discussed so far, they make double strand cuts. So I will take this one slide and then I will stop, okay? So there are the axis of symmetry, what we have discussed. This is for eco R1, right? GAA, TTC. All I need to remember for eco R1 or BAMH1 is GAA for eco R1, GGA for BAMH1. Because if you know this much part, the rest of the thing can be written, right? GAA, this, then you have to go with complementary to A and A, that is T, T, and complementary to G is C. Sorry, C is G. So either way, GAA, TTC, that's the sequence. And you make the opposite one, or the complementary one. And you should have known the axis of symmetry. I I am going to describe what it would be if it is R echo R1. I didn't have to mention R, but I just put it here. If you just mean echo R1, that means we are talking about restriction endonucleases. And what would happen if it were M echo R1? So GAA TTC 5 prime and 3 prime, you will have the opposite sides. Every time you are representing in your notes as a part of test or an assignment, you should always use the 5 prime 3 prime thing. Okay, so I will write the bottom one also, GAA TTC GAA TTC. Okay, so the slash here indicates that is the cut site. One of the subunits will cut here and the other subunit will cut here. Okay, I hope you are assuming that there is DNA here and here. So if I separate them, it would be something like G, C, T, T, A, A. That will be one part of it. And the other part would have been A, A, T, T, C, and G. That's the DNA. So this would have been the 5 prime and 3 prime, 3 prime and 5 prime. What would be the ends of this? This would have been the 5 prime. And here too, it will be 5 prime. This is 3 prime. This will be 3 prime. This part of the DNA is overhanging, right? It is the extra bit. If it were something like this, that would have been blunt ends, but you have this extra hanging that you refer to as overhangs. And this is the 5 prime overhang because it is the 5 prime side of it that is an overhang. And if you let it be, this part of the overhang is absolutely or perfectly complementary to this part. This A can base pair with this. The second A can base pair with the second T, and A will base pair with these two T's. That means they can form hydrogen bonds readily because they are perfectly complementary. Therefore, you call them as sticky ends. They're called as sticky ends for that reason. Okay. Last bit, and if you're adding M echo R1, the asterisk here that shows, that means they are methylating the A in that location. They methylate the 6 amino. You have a NH group, NH2 group that is on the adenine. 6 amino purine is adenine. So you have on this part, they would have added a methyl group. The same is the case. It is usually represented like this. This is what it means. Okay, I think I'm overshooting. So if you have any questions, you can ask. And the rest of the things I will continue in the next class. Any questions? Okay.
For now, no other doubts, sir. It's
Okay.
clear, sir.
Yeah, we'll meet in the next class then.
So
Okay.
I have a doubt.
Yes, please.
So you
Parvati?
mentioned that type 2, yes, sir. So you mentioned that type 2 restriction endonuclease is one of the most preferred ones because for experiment it gives you the certainty and the credibility as well. But
Yes.
in nature, in the terms of evolution, is there any predominant type of restriction enzyme that we could see? Like which RM system is predominantly seen, sir?
Yes. The problem is we don't know the origins of R&M systems. Hey, by the way, the others who have to go to the next class, you can leave. That's all right. But Parvati, I'm trying to answer your question. Is that in nature The origins of R&M systems we do not know. We don't know if it has originated within E. coli genomes, or is it from plasmids. Okay, so the prevalence uh, within within the bacterial genome does not have a bearing about bearing on the effectivity. But what I would think is the type 2 having two separate enzymes would be better for evolutionary purposes. Are you understanding what I said?
So could you say why? Because if it is together, maybe the transcription translation could be an easier process. It's like once
Yes,
initiated, both the yeah.
that's right. Say, for example, this is the site. Okay. If it is coming as one thing, R and M, which subunit is going to interact with this? Is going to change the consequence of it, isn't it? If it is methylation is going to happen, okay, this DNA will be protected from being destroyed. Yes. So
Yes.
that way, if you see in that light, for evolutionary purpose, here both the enzymes are coming to the same site at the same time. The probability of being cleaved or being meta-modified is almost equal. But instead, again, if you have the sites different, and if you have R and M separate subunits, and also the possibilities are like this. If you have more of these the numbers are more in restriction, and enzymes are produced in more in number compared to that of M, or the activity of R is more than M, then the probability of being cut is very high, isn't it? So it has to depend on concentrations of these enzymes, activity of these enzymes, and only then you know the consequence, whether this DNA is being cut or is modified.
Okay, sir. Just a follow-up question, sir.
Yes.
So you mentioned that if the concentration of restriction enzyme is higher when compared to the methylase or the modifying enzyme, and if the activity is also higher, then there is a more probability of that particular gene which was recognized to be cleaved than get protected against it.
Yes, that's right.
But but wouldn't this be true only when we start from time t0? Like, isn't it possible that the gene could have methylated already, like from like previously? So it does not. However, I mean, however bigger the concentration might be of the restriction enzyme, it will never have an effect because the DNA is already protected. Is
Yes,
that a
that is true
possibility?
for all genomic DNA. If you had a site, there is a methylation on one adenine and also methylation on one. And if it undergoes replication, it will become hemimethylated, isn't it? And everywhere there is hemimethylated, the methylase is likely to add more of these bases. We should have, there will not be any genome to start with as time 0, if you are saying. You will not have a genome consisting of these sites that can be recognized by the restriction endonuclease or methylase without any protection. Somehow something must have survived, and then the chain is continuously happening. After each round of replication, you will have this hemimethylation, and there is automatic protection from cleavage. The sites, even if they are hemimethylated, the restriction endonuclease does not leave those sites. Is that
Thank you.
understood?
Yes, sir. And one
How
other
this
doubt,
is
sir.
not, yes, yes.
Like how we have modifying enzymes, do we have, like, say for example methylases, do we have other, like, the demethylase enzymes also, like, so that this could be undone?
Okay, I mean, you're getting into epigenetics and all. This, what we are discussing, is purely about in prokaryotic systems. We can have demethylases, There are in epigenetics, if you're reading, you have denomethylases, methyltransferases, and also demethylases. We can use those to do it, but it's not, it's, say, if you have a methylated DNA, and you want to demethylate it, okay, we're talking about as a technique, okay? If you have methylated DNA and you want to demethylate, or is it easy to make a DNA fresh, the same DNA sequence? Can you make a unmethylated DNA? Right? There are two options. One is demethylating, and one is making a DNA without any methylation. This seems to be the easier one. So they would have made DNA without any methylation in general. Yeah.
But if we are speaking on a genomic scale, wouldn't it be like, if say I want this particular gene to be expressed and it has undergone a modification and therefore it is not getting expressed? So in that case,
the epigenetics in eukaryotes, right? You're talking, so in prokaryotes, the epigenetics is not as much. Methylation, demethylation, what you see in eukaryotic chromosome expression level. In prokaryotes, it's not that complex with regard to expression. They are much more direct. Okay, silencing of genes and other things are a little bit complex in eukaryotes, but it's not methylation dependent or unmethylation. Methylation is a rare thing in prokaryotes, unlike in eukaryotes. In prokaryotes, we have methylation, but that is for regulating the replication cycle, for example, or these kinds of methylation protection from endonucleases and so on. But certainly, if we want, we can do this. We can do it with CRISPR, right? If you have a sequence, we can make these spacers, and we can have defective Cas proteins, which with a conjugate of demethylases, which can then demethylate the DNA that is present here. That is a possibility, because you wanted a targeted demethylation. If you wanted to do that, that is a possibility.
thank you, sir.
Yes.
Yes,
But
sir.
you can
Thanks.
also get me. We'll discuss more at a later time if case you didn't clear it. You didn't get it cleared. Yeah.
Sure, sir. Thank you, sir.
Okay. Good. Thanks for the questions.